So in the last videos, we talked about how nucleotides are linked together in a condensation reaction to form a polymer of nucleic acids. And now that we have this single strand of DNA, what happens to it? So recall that living organisms must process or possess the ability to replicate and pass along heritable information. So how does DNA do this? And really, it's the secondary structure of DNA, which is a helix, that gives us this answer. So what is DNA's secondary structure? So early evidence, so before we really knew what was going on, suggested to us that the monomers were nucleotides, so we knew that. We knew that DNA had a sugar phosphate backbone, and a scientist named Chargaff established that the number of purines, which are adenine and guanine, remember nine atoms, the two rings, equaled the number of pyrimidines, right? The one ring cut, or CT, at least in the case of DNA. And that the number of thymines and adenines were equal to the number of guanines and cytosines. So Watson and Crick were around, and they were using models to try to figure out how these rules would go together. And Rosalind Franklin used X-ray crystallography to measure the distance between atoms in DNA. And this picture that she generated by using X-ray crystallography predicted a helical structure was the secondary structure. And as a sidebar, this is the X-ray crystallography image called image 51. And it was shown to Watson and Crick by Franklin's collaborator, Maurice Wilkins, without Franklin's knowledge. And so this picture really gave them the clue that this was a double helix and helped them figure out their model. So how do the two strands interact, right? So we had all these rules. And what Watson and Crick were able to work out were that there was this complementary base pairing that occurred, or this is often called Watson and Crick base pairing. And what this means is that guanine, right, is able to form a bond with cytosine, and actually forms 3 hydrogen bonds. So there's these electrical interactions, these hydrogen bonds, right, that are created from partial charges of oxygen and hydrogen that then allow these bases to interact. And so guanine and cytosine bond with each other, and there are 3 bonds. And adenine and thymine, right, also bond with each other, but there are only 2 hydrogen bonds. And these hydrogen bonds, right, that hold them together, right, hold the base pairs together and create a stable molecule. And because these hydrogen bonds are causing them to interact, they kind of pack in. And they're also held together by van der Waals forces, which are when atoms come together in this tight interaction, there's an additional attraction called van der Waals forces that holds them together. So this base pairing creates a very stable structure. Okay, so we have base pairing, right? And we've got our sugar phosphate backbone, right? And we've got our bases that point in this other direction of the sugar phosphate backbone, and the bases pair together. But for the bases to pair together, the strands actually have to run anti-parallel. So we talked about in the last podcast how we have 5' running into the 3' direction. So that's one strand runs in that direction, and the other strand actually runs anti-parallel. So it runs, if you read it from the top, it runs 3' to 5'. Okay, so it's just flipped over. The 3' hydroxyl now sticks up, right? Up here, it's still going to grow in that direction, but the 5' hydroxyl is at the other end. So this molecule, so it's said to run anti-parallel. So 5' to 3' and 5' to 3'. And this base pairing in anti-parallel direction forms the double helix. And so the sugar phosphate backbone faces the exterior, and the nitrogenous bases face the interior. And the hydrophobic interactions, right, of those base pairs, right, cause the molecule to twist and create a double helix, right? Double because there are 2 strands. And so here we just have the 5' to 3', 5' to 3' running in the anti-parallel direction, and we have our base pairing occurring between our nitrogenous bases, right? And the negatively charged phosphate groups that are hydrophilic are going to face outwards. The molecule is going to be hydrophilic overall, so it's going to dissolve in water, but it's going to twist to minimize those hydrophobic interactions. And so here, right, is the double helix of them twisting together. And this twisting or this helix creates grooves in the molecule. And so there's a large groove called the major groove, and then there's also a minor groove. And so this major groove and minor groove and the distance between the bases all reflect back onto that picture, image 51, right, where there was that pattern. And if you actually measure the distance between those bands, it actually exactly correlates with the distance between the bases, the distance between these grooves, and the distance between the sugar phosphate backbone. So that's why the image suggested it was a helix. So let's review a little bit. Sorry for this triangle right here, but we have our one DNA strand we talked a lot about in the last video, right? We've got our 5' running to 3', and we have, this represents our sugar phosphate backbone here, and this is just depicted here, right, with our carbons numbered, and we have this 5' carbon with our phosphate group, and then our 3' carbon with this hydroxyl group over here, and then we've got our bases facing that the inside of the molecule and the sugar phosphate backbone on the outside. And what happens is these bases then bond, right, via hydrogen bonds, and they're further stabilized by van der Waals interactions with another base. Okay, so thymine base pairs with adenine, and they base pair via these 2 hydrogen bonds, and adenine to thymine, and guanine to cytosine, right? And that has 3 hydrogen bonds between those 2 bases, and cytosine, guanine, again, 3 hydrogen bonds. And so these nucleotides come in, right, and they interact with their matching base pair, right? And then the phosphodiester bond is created, right? But it again runs anti-parallel, so this strand runs from 5' to 3' still, but in the opposite direction. And so that's how the double helix is established, and then that twists on itself. So here, right, this arrow here is representing a flipped molecule that's upside down. And we'll cover this in class. So a little bit of an activity preview. We're actually going to make a polymerizing strand, right? So we're going to add nucleotides in the right direction by flipping a paper over. So the tertiary structure, that was the secondary structure of DNA, this twisting, is actually is when it forms a more compact structure. So when DNA is wound too tightly or too loosely, it twists into supercoils, and it wraps around proteins. So in eukaryotic cells, those proteins are called histones. And so it allows the DNA then to twist on itself, and it compacts the DNA. And this allows for discrete units for cell division. It helps the DNA fit inside the nucleus, and it contributes to its function. And DNA, what does it do in a cell? It functions as an information molecule. So DNA can store and transmit biological information. It carries the information required for an organism's growth and reproduction. And it's the language of nucleic acids contained in that sequence of the bases that allows all of this to occur. So the nitrogen bases function like an alphabet, and those sequences have meaning, like the letters of a word. And so different sequences can code, they're copied into RNA, and then that RNA, the sequence of that RNA is then used to make a particular protein. So DNA, as I talked about, also, kind of as I hinted at the summary, serves as its own template for synthesis of a complementary strand. So remember, the characteristics of life is that we have to be able to replicate itself. And so the way this is done is through DNA. So it contains all the information required to copy itself, an exact copy of itself. And in modern cells, this process requires enzymes. And so what happens is you have your DNA double helix, and so the 2 strands are separated. And so if you take one strand, right, running from 5' to 3', that's the template strand. And just as I talked about in the summary, right, those different nucleotides are going to come in and make hydrogen bonds with whatever base pair they're supposed to interact with. So if you have a G It's a cytosine is going to come in, and it's going to interact, form hydrogen bonds with a guanine. And if it's a thymine on your template strand, an adenine is going to come in. And so you have those hydrogen bonds, and then that sugar phosphate backbone is going to grow in the 5' to 3' direction, but right anti-parallel. So it's going to grow up. And it's going to create a new strand. So you had your template strand, and then you have your new strand, and it's going to create the double helix, and now you're going to have 2 molecules of DNA. And so that's how DNA copies itself. DNA is very stable, it's resistant to chemical degradation, and because of that, it makes it a really reliable macromolecule for genetic information storage. And stable molecules like DNA actually make very poor catalysts. And so DNA has never been observed to catalyze reactions. So biologists do not think that this is the first life molecule, right? That's thought to be RNA. We'll talk a little bit about the structure of RNA. So RNA, like DNA, has a primary structure that consists of that sugar phosphate backbone that's formed by phosphodiester linkages. And like DNA, it has 4 types of nitrogenous bases extending from it. But the primary structure of RNA differs slightly from DNA. And that's because RNA contains ribose instead of deoxyribose. And as we pointed out in the first lecture, the ribose has a 2' hydroxyl group, and this 2' hydroxyl group is more reactive than the 2' hydrogen in DNA. So RNA is much more reactive, and RNA is also much less stable than DNA. Another difference of the primary structure is that RNA contains uracil instead of thymine. So those are some differences in the primary structure. And the secondary structure also differs compared to DNA. So RNA does form a secondary structure that results from complementary base pairing, so A with U and G with C. The difference is that RNA usually forms this structure called a hairpin. So what happens is you have a single strand of RNA, and you have complementary base pairing occurring between a strand that essentially turns over and then wraps around on itself. So complementary bases pair between anti-parallel regions of a single strand. So this structure here can contribute to some of the functions of RNA. So RNA can also have tertiary structures that can form more complex shapes. Their shapes are more diverse in size and shape and reactivity than DNA. And this makes RNA highly versatile. It is an information-containing molecule like DNA, but also is capable of self-replication and capable of catalyzing chemical reactions. And so your textbook talks about ribozymes. So you should definitely, if you're interested in this, read more in the text. So RNA is capable of self-replication. This information stored in RNA can be used to make copies via complementary base pairing. And it must pass through double-stranded intermediates. And this doesn't happen in our cells, but it occurs in some viruses. So just to summarize, right, we've got our DNA. We've talked a lot about its primary structure, right? You have these 2 anti-parallel strands that come together with complementary base pairing, and they wind into a double helix. And then the tertiary structure, right, it supercoils and it wraps around protein to kind of compact itself. And then RNA, right, you've got your same backbone. It doesn't form that double helix. It does form this hairpin structure, right? It's just one strand that has some base pairing with itself. And then it forms these complex secondary structures, and it can have catalytic functions. And it's really thought to be the first life molecule. So that's everything I have for today's videos, and I look forward to seeing everyone in class. Thanks, bye.